Welcome to AgriTalk and thank you for keeping it KTN Farmers TV. On today's show, we are talking about plant biotechnology, and with me in studio is Professor Richard Oduor, who is the Director of Research Support and Dissemination from Kenyatta University. Welcome to the show, Professor.
Thank you, Philip.
Tell us a little bit about yourself and what you do at the university.
I'm a professor of molecular and cell biology from Kenyatta University. I also chair the Kenya University Biotechnology Consortium, but currently I sit in as the Director of Research Support and Dissemination at KU.
Okay. When we talk about plant biotechnology, what is it exactly?
Well, there is no really overarching definition, but if you talk about biotechnology in general or plant biotechnology, then you are talking about any technique that uses living organism or living systems or products from those living organisms to develop products that can be commercialized or utilized by organisms or just any living system. So yes.
So when is the origin, or when did we start the first time the human being started using biotech?
Interestingly, Philip, what I find interesting is that this thing started way early. Before, if you remember, we were hunters and gatherers. And therefore, what that meant was that you walk whatever distance you can cover and run around the field. If you find something that is edible, you will pick that and enjoy it. Then you continue hunting. But I think the moment human beings decided to be a little lazy and said, no, 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 no. Why would we keep walking? And these plants, we know them. The moment they started now to plant them so that they don't walk all those distances, because when you start walking and you're a hunter and gatherer, you don't know exactly where you will get the next mango or the next banana. They're all in the field. But because repeatedly they got used to particular plants and sources of their food, they decided, why are we wasting a lot more time? Why can't we just have these seeds growing around this particular area and we can harvest them a little easy? That is when it's all started. And of course, they have been using biotechnology, but they didn't probably know what it exactly meant, because when you talk about fermentation, when there were, milk, and probably you are well more sick on many of these things. That was biotechnology, and it has not started now. That is something that has been with us here for several years. So it is as old, if you ask me, as the time when we became lazy and started using technologies. It's the simplest I can say.
Okay.
But of course, we've got other detailed definition from my field, which might not.
Okay, apart from fermentation and all that, which other benefits can we accrue from biotech?
More. If you talk about plant biotechnology, and probably we have a number of things, including tissue culture. You can talk about drug discovery. And when you talk about tissue culture, for example, we talk about say, tissue culture banana. There are reasons why we do tissue culture, so that we make those planting materials a bit clean. Because the disease, remember, for example, when you talk about banana, there is no seed that you're going to plant. It means you'll uproot the sucker and then go plant it. What that then means is that if that banana had some viral diseases, then you have just plucked that disease and taken it to another place as well. So in tissue culture, we have a way of how we can clean the planting material so that at least that cycle of that virus is stopped from being recurring even to the offsprings. And that's why we subject them through the tissue culture, so that we have a clean planting material. We do that a lot more. That is tissue culture. But in drug discovery, we also have a lot of how we have used biotechnology, modern biotechnology that is, to generate drugs. And probably we'll go deeper into that as time goes.
Okay. Are there more examples, or can you give us more examples apart from tissue culture that
Yes.
we can expound on?
Yes, I can. In medical field, you talk about insulin. Any person that you find out there that has diabetes, that person right now is enjoying GMO insulin. Why? Because before, you see, how was insulin actually made? It was made from pigs, first of all. And imagine the number of pigs you need to kill for you to get enough isolates that would now be used for human beings. That's number one. But at the same time, there were challenges in terms of religious, challenges. Look at a Muslim who is diabetic to enjoy insulin that has been, say, purified from a pig. Clearly, you're allowing those guys just to die because that is religion, it's their faith. So then that is when biotechnology came in and said, look, why can't I get into a human being who already has the right insulin, identify that gene that is responsible for to produce the real insulin, then take it and then clone it into using recombinant DNA technology, which is just the basic of, word of just cloning. Put it into a bacteria and allow that bacteria, which is not disease causing actually, allow it now to be producing for you insulin as you purify, just like an extract. That is what we are talking about right now, and that is what biotechnology actually is doing. So we are no longer killing pigs, and no person can say I don't want it because of my faith, and yet we are also not actually, it's not expensive as such, so you can scale it up. Because even if you chose that pigs would be the only source of it, how many acres of land do you require to rear the entire pigs to even satisfy the entire world? But these are now things that we produce in industry, just like how we make Coca-Cola and all these things. So that's what we are calling industrial biotechnology. Yes.
So you talked about GMO, genetically modified organisms. It is one name or one word that most people don't want to hear around here. Tell us a little bit about what GMO is and why is it that we fear it that much.
Yes, I'm glad, Philip, that you've asked that question. See, when you talk about GMO, it is clearly misunderstood. And probably, and I take responsibility sometimes as researchers, because over the years we haven't been talking about it a little bit more, at least to break it down for the intended consumers. Genetically modified organism is nothing new. What it only means is you are taking a gene from one organism and moving it into another, only if there is benefit. And let me go a little basic. If you are eating githeri, which is a common delicacy for many of us, but there can come a time. Right now, we are not saying it is yet time because beans are still growing naturally, right? And therefore, there's no need. But in the future, if assuming there is a disease that can come and clear the entire or make it very difficult for beans to grow, what are we supposed to do? Because we still require proteins. So what we do as genetic engineers, we will go into the beans genome and say, identify for us what are the section of the genes that are responsible for only the proteins. Because we don't want the roots. We don't eat the roots. Or we probably don't eat the stems, all right? But we want the protein that is in there within the bean seeds. And because the genome of these plants have already been sequenced, we can clearly get to know this is the section that is responsible for the root, this other section is responsible for the leaves, and this other section is responsible for the stem. So you go in there and identify the genes. Once you picked that gene, that is the gene that now, those are the genes that now I transfer into maize. And when I take them to maize, because we also have the genome of maize already sequenced, what do I do in this genome? I put these genes in here, but target that protein to be expressed in the kernels because that is what we eat. You don't want the roots to be more keeping your proteins because human beings don't eat the roots. So how will I target that? We have techniques right now of getting to know how to target where that gene is expressed. You ask yourself why your fingers are not on your head. Why? It is because there's something that guides the spatial distribution and expression of these things that you see. And that is now why when we talk about genetically modified, at that particular point, you have just moved the genes that naturally you eat them anyway in beans. Because I don't see you when you're eating beans and when you're eating, githeri, you go into the genome and start plucking the nucleus so that we are mixing them. And the fear that people have is that then I ask you, why don't you wake up with sounding green all through? Because you have been eating the total genome with chlorophyll in it. And if you eat cows, you eat it together with the horns, and we've been eating them for several years. What happened to that DNA that you've been eating in the beef all the time? And why do you now start thinking that it is really so bad if we now say that instead of, because cows are walking several distances that are really not, we don't even have the germplasm. Why can't we take the gene? If we have goats that grow better and we need the proteins that probably is from another animal, just identify the gene itself. Move it to this thing that still survives, that the environment has still allowed to survive, so that you can still produce. Genetically modified organism simply means that we have picked genes from two different organisms that naturally do not cross. Okay? Because if you, for example, say beans and maize, you cannot cross them. But genetically, you are able to cross them. So it has gone beyond what naturally we call the species barrier. At least genetically, every other thing that is alive out there, by the way, only have 4 letters. It has only 4 letters. Anything alive, mangoes, human beings, anything that is alive out there has only 4 letters. Guanine, adenine, tyrosine, and adenine. Those are the only 4. The only variation why you are not looking like a pig and not like a cow, and yet we only have the 4, is how they follow each other in what we call the sequence. So that is the number of how many and how they follow each other. But apart from that, genetically, anything that is alive is a relative. It's a relative, and that is what now genetic engineering has helped. That you see a mango tree and banana tree, and you feel that you need to get the vitamins that are more in mango tree and are not in banana. Then you go into the genome where there is no species barrier, and then you pick it carefully and you move it to the right place. Because what is it? What is the worst that would happen? Mango naturally exists, banana naturally exists. So does it just become bad because now you have mixed them? Yet you take that juice that people like and they call it cocktail. So you can see the simplistic bit of the things.
Yes.
Yes.
So I'm trying to wonder. So where did this all language of it is bad start from?
And that is why I started by saying I take responsibility on behalf of all the scientists, because we haven't been talking. We do these things, they are good for the intent, we have very good aim and purpose for doing these things, but we never articulate them a bit simply for the people who are supposed to consume them. But also there are also general concerns, uh, like because I am able to do genetic engineering, I can comfortably edit even the human beings and transfer the genes. But is it ethical? Because if naturally, we only have, for a human being to be born, we only need a sperm and an ovum, right? Then you mix them. It becomes a zygote, fertilization. After that, how the head happens, how the legs, how other cells divide into becoming blood and the eye and other thing is left for the system. And because those systems are already known, they are developmental stages, we can comfortably, actually through genetic engineering, be able to pick a cell and with that cell regenerate the whole organism from it. Like if I want banana, I don't need to now wait for the banana or mango tree to grow until it bears for me the seeds, because that might take several years. I just go onto the tip of it, just pick a small leaf and go into the right medium in the lab. I will be able to get the whole organism. So the thinking would be, somebody would stretch their imagination and say, how about if I come and pluck your hair and just get the gene and take you into the petri dish and try to regenerate a human being like another Philip? While that is very interesting and I would love to do that, is it really ethical? And who now does this new person actually pray to? Because I would insist that pray through me to the other person who gave me intelligence. So there are quite a number of things that are happening, and because of that, there are a lot of restrictions on and parameters on what you can't do and what you can do. So it is self-regulated.
So, Prof, we'll go to gene editing later on.
Yeah.
It's one of my topics that I've been trying to research in the recent past. Let's keep a little bit on the benefits of biotech, especially for us in Africa, that we keep complaining of hunger all the time. How can we use this technology to produce adequate and healthy foods for this growing population, having in mind the changing climate and global warming?
Thanks, Philip. In a lab that I had at Kenyatta University, which is the Plant Transformation Laboratory, before I moved to become the Director of Research at Kenyatta University, that is the only lab in Eastern Central Africa in the universities that is certified and accredited to do genetic modification. There are only 3 such labs in this country. one in Calro and another one in Il Rebecca. And the KU one was the first one that has been there since 2005. So we are talking about 16 years. So for 16 years, we have been doing genetic modification in this very, very country with Kenyans and training them, and they have done a lot more. Now, to be a little bit simplistic, drought is one of the challenges that we are encountering now. And and as we are talking right now in the lab, we have drought tolerant maize that we have made here through the funding from international community, and also specifically sponsored by or supported by Kenyan government, NRF. They started from almost 2012. We have had funding every single call to support genetically modified organisms. And why do we get this funding from the government? And that is why probably you will now start asking, is the same probably government that doesn't want the GM, and how come that it's sponsoring again or supporting research? Even as we are talking right now, we just concluded another research, which there will be another documentary very soon on it, by NRF. We were trying to make drought-tolerant maize. We were trying to advance research on cassava. Look at cassava. It's very bitter sometimes. Yeah. So the bitter cassava is because that cassava is trying to survive. There is a reason why, because if it becomes sweet, then you eat it so very quickly and it will have a lot of attack. So don't ever forget that these plants actually are not aware that we use them for food. If plants will ever know, and this is a secret we need to keep, is that maize will seriously, if it realizes the cob that we pluck and we go eat, it will start producing the cobs on the root so that nobody sees, because it is about survival for the fittest and it's about existence. Now, cassava produces cyanide. That bitter thing is called cyanide. It's a defense mechanism so that it's not overly eaten.
That is the reason we've had a few people die for consuming cassava.
Consuming cassava. There are different varieties, others with a lot more. If the concentration is more, it's better for that cassava because it won't be liked. So it will survive forever. And you will notice those that are usually bitter have very serious, big, sizable tubers. The ones that are very sweet are known, and we have propagated them, and they are usually they will be subjected to extinction if we are not careful. But because we have enough land and all that, we can still keep them. But the reason why I'm bringing this up is, how do I come in now as a researcher? So in the lab at Kenyatta University again, we have identified what is this pathway that leads to the production of that cyanide. So through genetic engineering and RNA gene silencing, you are able to reduce that enzyme, its activity, from producing that cyanide so that that cassava is still edible, so that it produces a cyanogenic cassava. That is already a product we have here at Kenyatta University, sponsored by, again, funded by Kenyan government. Maize that I'm talking about is drought, against drought. We have developed maize that is tolerant to drought stress. Now, how have we gotten this? You see, when a plant senses that we have drought, the first thing that a plant does is to shed off the leaves. Why does it do that? Because the leaves are where there are those holes that we call stomata that will allow loss of water a lot more. So by dropping all of them, it means therefore that the plant is a little safer in terms of transpiration and loss of water. Now, that is good, but also bad at the same time, because you also require the same leaf to be the source of photosynthesis so that the plant can also give you a bit of yield. So what have we done? We have identified what is the gene that can make the plant to be a little tolerant so that they don't shed the leaves quickly, to allow them produce for you some yield even when there is drought. And also another one to elongate the roots. Because if you have a plant that has got 100 meters of roots going down, that plant will never suffer from any lack of water because there is enough water in that distance if you take it down. So it means if you look at the roots for maize, they are not as deep. But if you look at other plants, they are deeper. So we have also identified other genes from other plants that have deeper roots, so that we make sure that the roots are deeper and the leaves are also not necessarily lost, or what we are calling reduced, or senescence. Senescence is that bit of losing the leaves when there is a bit of drought. So what we have done is we've combined that, and we are also producing maize that are drought tolerant.
We'll take a short commercial break, but we'll be back in a few.